My name is David Keator, and I'll be hosting this training session today. It's kind of more of a training and hacking session. I've posted in the chat window a link to the Google Drive folder, which contains the agenda for the different parts of today's training and tutorial, and also then links out to data files we'll be using in each of the sections and slides that will be presented. So if you are able to go to that link and you click on the part 1 document, you will see the what I have on the screen essentially, which is an overview of this session and how it will go. Again, I'm David Keator. I'm from the University of California, Irvine. We'll start out, excuse me, with approximately 50-minute, these times aren't going to be set in stone, but approximately 50-minute introduction where we'll have some speakers present some slides and give you some information and context into what is going on in sections 2 and 3. We'll have, besides myself, we'll have JB Poling from McGill University, JB wave. There he is. We'll have Keator from UC Irvine. Hi, Nazek. Yeah.
Hello.
All right, and we'll have Theo Van Erp from UC Irvine as well. Hi. The wave. And so then, so we'll go through the presentations, then we'll take a 5-minute break, and then we'll start on part 2. Part 2 is more of a practical session where we'll use some of the NIDM tools and a use case from the Enigma Consortium and do some data annotations and create some NIDM files. And we'll show you the nice things you can do with those files in terms of querying and some other interesting bits. Also, training you on how to annotate your own data to use some of these techniques to, for example, if you have a bits data set, create a bits sidecar file. And then we'll, in part 3, we'll give you an opportunity to annotate some of your own data or make some JSON sidecar files of your bits data sets if you choose to do so. And then we'll wrap it up. We should make it within the 3 hours. We may be a little early if there's, and so we'll just see how it goes. If there's any questions, raise your hand or ask a question in the chat window, and we'll, between Nazek, Theo, JB, and myself, we'll try to catch the questions and try to field them. We'll also take questions at the end of each of the initial presentations, as opposed to waiting till the end. That way, if there are any questions, we can get to them when the presentation's fresh in your mind. Okay, any questions, any other comments from anyone before we get started? All right, so I will flip over to the slides for first set of slides. Here's me. Okay, so there's always a question. On the screen, do you see the presenter view or do you see the slide view?
I see David only and no slides.
You see no slides. Okay, so hold on 1 second. Share the screen. All right. How about now?
They're not in presentation mode, but they're on the screen.
So it's full screen mode. Is that right? Can you see the slide in full screen?
Yeah, the PowerPoint presentation is full screen, but it's not. Okay. Sorry, it's not presentation mode. It's I'm seeing the slide as if you could edit them.
Okay. All right. 1 more second here. Sorry about that. This happens because I'm using multiple screens. Start share 1 more time, and it should be fixed. Okay, you can just see the slides now, right? All right. So I will have to squint. Okay, so today I'll be briefly telling you about some of the tools and techniques for annotating both BIDS data sets and tabular data files, also adding in brain volume data, this very sort of human neuroimaging focused, but you can use the tools for other purposes. So I'll be telling you a little bit about this, and then in part 2, you'll be seeing some of this done live. So hopefully between the two, you'll have a appreciation for how this works and what we're doing. This work has been supported by the National Institute of Mental Health, Fair Data Descriptors Grant, and NIDM under the ReproNAM effort. So just many of you know NIDM or have heard of it, have seen this slide many times, but just real briefly, essentially the neuroimaging data model, what we call NIDM, is just a set of specifications for how we link different components of the scientific workflow together, or our attempt to do so. It's composed of 3 main components: the NIDM experiment component, which is what we'll mostly be seeing today, a NIDM workflow component, which is aligned with some of the BIDS provenance work that's going on in one of those BIDS extension proposals for the provenance, and NIDM results, which is there's a couple of tools out there for SPM and FSL created to capture, mass univariate statistical results in using the same sort of tools and techniques as capturing, in this case, experiment metadata and hopefully in workflows. The idea being you'd have like a consistent syntax and a consistent way of describing the life cycle of data right up to publication. The NIDM is built on using semantic web techniques and builds off the PROV family of specifications, and has a vocabulary of how we say things about neuroimaging data. And so by building off of the semantic web technologies, we get for free a rich query language and the ability to essentially link bits of information with the wider scientific community, and also within the document itself, making these documents, we hope, unambiguous and semantic, self-annotating, such that if you get one of these documents sometime in the future and the person who generated it has left, you'd be able to, from the URIs and the way semantic web style documents are set up, understand what's in it. So the NIDM experiment model is quite simple, and we think pretty intuitive. It's a hierarchy. Essentially, it ends up being a graph, but at the highest level, you have this project activity, which can contain as much metadata as you like, as long as it's formatted according to some semantic web rules, where there's a subject, a predicate, and an object. And this project activity, there are many sessions that are part of a project, one or more sessions, and then there's these acquisition entity pairs, which is where the actual data would be stored. An acquisition activity could be an MRI acquisition. It could be an assessment acquisition. It could be an EEG acquisition. It could be any kind of acquisition, really. And there's an activity surrounding that kind of captures the provenance of what was done. And this box that says acquisition activity could be itself an entire provenance graph, but right now it's just a box with some metadata. And then the entity contains the data or information about the data, also formatted according to the semantic web rules. And so from that simple hierarchy, you can create fairly complex graphs. Here on the left side, we have the NIDM experiment portion of the graph, which has the project sessions and acquisition hierarchy, where on the left we have some acquisitions that are assessment acquisitions, and on the right we have more image acquisitions that may have metadata about the images themselves, right down to links if these images are available on the web. And then on the right side, the very far right of this slide, we have some results. These are from FreeSurfer and basically computations of volumes of different subregions of the brain. And we'll show you some of that in the later sessions. And these are linked to the images that were used during the segmentation of the data. This is the, in this example, you could build this out and have more and more different types of analyses linking to the original data that was used, and then have the provenance captured. So with the NIDM, in the NIDM toolkit, we have a Python-based toolkit called PyNIDM. Inside that toolkit, and there'll be some links in the part 2 to download that. I think it was also on the training space announcement. There are a variety of tools. We will touch on most of these today in part 2, but essentially you have some annotation tools that take either a BIDS data set and allow you to annotate the tabular data contained therein and create those sidecar files. And as a output to this process, you also get a NIDM record describing that entire BIDS directory. And with the query tools, you'll see why that could be useful for you. You can also, again, once you have those NIDM files, you can add additional assessment data to those existing NIDM files linked by the subject ID using the next tool, CSV to NIDM, which will take any CSV or tab separated values, TSV file, and allow you to annotate that, create a data dictionary, also add that information to an existing NIDM file. And then there's the last tool that we'll touch upon today with respect to this kind of thing is the segstats to NIDM tool, which allow you to point to a FreeSurfer directory and write out those FreeSurfer, volume measures and surface-based measures into an NIDM file, or add it to an existing NIDM file linked by subject ID. And then we have a query tool, PyNIDM query, which has a variety of both parameterized, like canned queries, and then also the ability to build up queries with a REST-style API. Also accepts Sparkle queries in the form stored in text files if you're familiar with Sparkle for RDF. And then we have a variety of utilities to convert NIDM files from our preferred RDF format Turtle to JSON LD and some other RDF syntaxes. We have a concatenation tool where you can just add NIDM files together. We have a merge tool, which merges NIDM files based on subject ID. And we have a visualization tool, which lets you just really see the graph itself as a, as an image, as a PDF document, essentially. Okay, so if we were to run the BIDS to NIDM tool, everybody probably is familiar with BIDS at this point. The left side's a DICOM, standard kind of DICOM data structure that you might get off your MRI machine. On the right is the BIDS structure where you've renamed some files and put them into a hierarchy with some semantics that, anatomical scans are stored under a NAT, functional scans under func, so forth. And when you run the BIDS to NIDM tool, what it does is it'll both evaluate the entire structure of that BIDS directory, but it'll also open up the participant's TSV file. It'll iterate over the header row. It'll ask you to annotate each of the variables so that we're capturing properties about units, about ranges, about data types, and so forth. It'll output that sidecar file into your BIDS directory for you. And so then you essentially now have an annotated BIDS data set that can be useful for query. It'll also give you a NIDM file out from this process that you can use with the RDF query tools. And you can choose to have those additional files like the NIDM file added to .bidsignore. That way it doesn't mess up your BIDS data set. It's still a valid BIDS data set. So how do we do this annotation and creation of these data dictionaries? A schematic is shown. Essentially, we can take the BIDS images or the assessments. They get loaded into these NIDM annotation tools. We basically do a search of a variety of information resources to allow you to link concepts to your variables, and you'll see some of that later. That makes it easier for us to query across data sets, but it also gives you the ability to then add your own data dictionaries for the variables contained within your files. and then uses all that information to output the various NIDM file and the JSON sidecar files. So here's an example of a participant's TSV file from the ADHD 200 data set from the Brown site. This publicly available data, you can see it as participant ID, gender, age, handedness, verbal IQ, and so forth across the top. Those are the variables contained in this file. Essentially, what you do is you run this bids to MRI to NIDM tool. You give it a path to the bids directory. And in this case, I'm telling it to add the output to the bids ignore file. It then found that gender. So it iterates through the participant.tsv file, finds the first variable gender. It asks you to put in a definition. This will all happen in part 2. You'll be able to see this done with the Enigma data set, essentially using a CSV file, but the process is exactly the same. And it's just capturing for your categorical variable gender. The 2 categories in this case was just male and female. The second part of this here is to choose to associate a concept with the variable. If you choose to do that, the idea here is that a high-level concept describing what gender means without units and without your particular coding will be linked to your variable. That means then that we can query across data sets and just say, give me all data sets that had a measure of gender, and we don't care exactly what you called the variable where you stored gender, and we don't care at this level if you're coding the male, female, other, etc. 1, 2, 3, it doesn't matter. We just want to find data sets with gender and age, and that's why we do this concept association. Not all variables need to have a concept associated with them. In fact, we prefer that all variables don't. The idea here is we're trying to make querying easier across data sets by this optional concept association. And so once you do that, the JSON file that comes out looks similar to the one shown here, where we've captured some values like minimum and maximum and the data type or value type of your variable, and categorical variable mappings to numbers if they exist. And then through this is about property, we've captured a concept that's been associated with these particular variables. That can live with your bids directory, and you can use it with regular, any bids tool. It looks just like a bids sidecar file. We have another annotation tool that's in development you'll see as well today. But I don't think it's ready for prime time yet, but it's a JavaScript tool to allow you to do this whole annotation process graphically as opposed to on the command line. And then once you do all these concepts, you associate concepts with your data sets. This is an example where we went through OpenNeuro, the database, and went through each bids data set, and we went through the variables contained therein, and we associated concepts with selected variables from these OpenNeuro data sets. Now, what we're doing is we're building a concept-based query where we're saying we just want to see data sets, in this case, that have a measure of anxiety disorder, a measure of age. And when we do that query, we find that there's 3 such OpenNeuro data sets that have measures of anxiety and age. In order to determine what those measures or the measure of anxiety was, for example, you would have to dig down further into those sidecar files, which is why they're so important to understand exactly for that data set, what was the measure of anxiety disorder, how is it coded, and can I combine different 2 data sets together and use them. So once you do all this annotation and you create your NIDM files, you could then use PyNIDM query. It has the Sparkle query and support for Sparkle queries, support for a REST style query engine, and allows you to also run a Docker hosted graph database locally, which improves some query speeds. We'll see how to use this tool in part 2, but it has a lot of parameterized queries and then allows you to do some other things in terms of querying. And so just real quick, if you were to run PyNIDM query and give it that NIDM file that came out of your bids example and ask for which participants are included in that data set, you would get a list like this where there's a unique identifier on the left that identifies the node in the graph. And then on the right was the study-specific participant ID. You can also do a data elements query, which shows you all the different data elements that were collected as part of the study. And so we'll see a little bit how to use that later. And lastly, the free surfer tool, which you'll also see, SegStats to NIDM lets you provide either a subjects directory from the free surfer segmentation process, so the output, or just the APARC ASEG file, or a CSV file, which we'll see during the Enigma section, how you can use a CSV file that contains free surfer derived brain volumes and add that information to your NIDM file. And so once you do that, if you choose to add the brain segmentations to the NIDM file, it searches for your subject ID in the NIDM file and then associates the brain volume data with it. In the NIDM file, you end up getting a bunch of volume data that's associated with a free surfer common data element. These are elements within free surfer that free surfer measures. And there's a data dictionary essentially associated with those as well, which can be used during the query process. And that's all I have for my section. And I'd like to thank all the people who helped work on this. So let's see here. So are there any questions or anything in the chat window? If not, we can move on.